neurons, neural circuits, and brain regions for vision. A human brain has billions of neurons. This picture of a neuron is drawn by a pioneer neuroscientist, Cahal. Soma is a neuron's cell body. Dendrites of a neuron receive signals from other neurons or sensory signals from outside the brain. Axons of a neuron send signals to other neurons, muscle fibers, or other effectors. Typical outputs from the neuron are in the form of electrical pulses called action potentials or spikes. A neuron's state can be briefly described by its membrane potential relative to the extracellular media, and this largely determines the neuron's propensity to fire action potentials or to spike. A synapse is a connection between neurons to send signals from one neuron to another. Here is a simple model of a neuron. U models its internal state, for example, its membrane potential. GU is a function of U, it models its output, and often this output is described by the rate of action potentials fired by a neuron. And GU is a non-negative function, it increases monotonically with U. It usually involves a threshold for the membrane potential U and a saturating output level. I is the input or input current injected into a neuron. Often it is from other neurons' outputs through synapses. Given a neural connection synapse, and you have the postsynaptic neuron and the presynaptic neuron. Driven by input I, a neuron's state U changes with time t, or evolves with time t, and this process is modeled by this equation. In this equation, tau is the membrane time constant of the neuron. A typical time constant value is 10 milliseconds, for example. So, for example, plotting U as a function of time t, if U starts at 0 at time t equal to 0, and an injecting input I starts at this time, then you will rise with time and eventually reach to a level U equal to I times tau, and it takes time duration tau to have most of this state value change in U. The action potentials are actually fired at random times, so GU is actually the average number of spikes fired per second. And let's use R with a bar over it as the average spiking rate, and we call it R bar. The actual R, that's the number of spikes during a particular second, follows a probability distribution, and often this distribution resembles a Poisson distribution, and this distribution has an average R bar, and it's like a Gaussian or normal distribution when R bar is large enough. A brain area can be modeled as a neural circuit containing many interacting neurons. It receives input from some brain areas and sends output to some brain areas, and often two different brain areas can be reciprocally connected with each other this way. Here is an example model circuit, and this one is of the primary visual cortex, also called V1. Neurons that do not send outputs to other brain areas are called interneurons. And a neuron is an excitatory neuron when its outputs excite other neurons. And it can be modeled as injecting positive currents into other neurons to make them more active or to increase their chance to fire action potentials. A neuron is an inhibitory neuron when its outputs inhibit other neurons to make them less active. In our brain, a neuron cannot be excitatory to some postsynaptic cells and be inhibitory to other postsynaptic cells. In other words, a neuron is either exclusively excitatory or exclusively inhibitory, and this is called Dale's law. Typically, only excitatory neurons send outputs to other brain areas. So inhibitory neurons are almost always interneurons. Most neural connections in the brain are local, so neurons are more likely to be connected to other neurons in the same brain area than with neurons in other brain areas. Here is a monkey's brain. Monkeys' brains are often used as models to study human brains. This is a view from this brain's right side. This is the eyeball, this is the front of the brain, the back of the brain. If you unfold and spread out the cortical sheet, you have this unfolded brain. This is a retina for one eye and for the other eye. Visual inputs go from the retina to the primary visual cortex, also called V1. Then V2, V3, V4, IT, V5, also called MT, LIP, etc. And these colored cortical areas visualize regions devoted exclusively to vision in the brain. Information flows from visual areas to frontal brain areas. Here is FEF for frontal eye fields. Here is SC for superior colliculus. It is an important brain region for making eye movements. Many brain regions project to this area, including particularly retina, V1, frontal eye field, and LIP. Here is a schematic of visual processing hierarchy. V1 is also called the striate cortex. The collection of brain regions within this gray box is often called extrastriate cortical areas. Many connections between brain regions are reciprocal, so the lines connecting two areas in this schematic, many of them often have no arrows for marking a particular direction of signal transmissions. For example, between V1 and LGN, there is a feed-forward of visual inputs from LGN to V1, and also a feedback of signals from V1 to LGN. And this is also true for between V1 and V2, and between V1 and V4, etc. So many connections between two brain regions are reciprocal for visual processing. This is how electrophysiological experiments measure what visual inputs on the display screen can make a neuron fire, or to make a neuron fire more frequently. Each neuron typically responds only to visual inputs in a limited region of the visual space, and this limited region is called a neuron's recidiv field. For example, this could be the recidiv field of one neuron, and this could be the recidiv field for another neuron. Size of visual space or recidiv fields are measured by the degrees in visual angle, and 1 degree of visual angle is roughly the width of your thumb at 1 arm's length. The sizes of the recidiv fields increase along the visual pathway, so its scale is about 0.06 degree for the central part of the visual field for retinal neurons, but for neurons in IT, the scale of the recidiv fields is about 20 to 50 degrees. Much more is known about the recidiv field properties in the retina and V1 than in higher visual areas. In the 1950s, Stephen Kuffler discovered that visual input shapes to excite retinal neurons are isotropic. They are more or less dots, such as a white dot in a black background, or a black dot in a white background, or a red dot, a green dot, etc. One dot for one retinal neuron's recidiv field. Collectively, the different recidiv fields from different retinal neurons tile up the whole visual field. Soon after, Hubel and Wiesel discovered that neurons in V1 are excited by shapes like small bars and edges, black bars, white bars, red bars, green bars, vertical bars, tilted bars, etc., and even bars of particular moving directions. However, in higher visual areas along the visual pathway, residual field properties are not only more complex, they are also more difficult to nail down. But these are some of the shapes observed to excite neurons in V2, V4, IT, etc. Compared to situations in V1 and retina, the neuroresident fields in higher areas depend on whether the animal is awake or anesthetized, and depend on what the animal is paying attention to. Here are the residual fields from 17 neurons in V1 of a monkey. They were recorded by a single penetration of a recording electrode. Each residual field is marked by a rectangle. For example, this is the residual field for the 14th neuron, and this marks the size of 1 degree of visual angle. So this residual field for the 14th neuron is larger than 1 degree in both its width and height. But some of the residual fields have a scale smaller than 1 degree. This cluster of residual fields are about 10 degrees from the center of gaze. Here are 3 clusters of residual fields in monkey V1. This is the center of gaze. This cluster of residual fields is about 1 degree from the center of gaze. A second cluster about 7 degrees from the center of gaze, and a third cluster about 10 degrees from the center of gaze. Generally, residual fields are smaller in the central visual fields and larger in the peripheral visual field. And this is partly why our human visual acuity or spatial resolution is better in central vision. The visual pathway is also divided into brain regions belonging to the ventral visual pathway versus the dorsal visual pathway. Information about what visual objects are tend to be more in the ventral pathway, and information about where visual objects are tend to be more in the dorsal visual pathway. By highlighting the circuit components for gaze control, this schematic also emphasizes that vision contains both looking and seeing, and ultimately vision is for the purpose to act onto the world.